Antibody C regions confer functional specialization. The secreted antibodies protect the body in a variety of ways. In some cases, it is enough for the antibodies simply to bind antigen. For instance, by binding tightly to a toxin or virus, an antibody can prevent it from recognizing its receptor on a host cell. The V regions on their own are sufficient for this. The C region is essential, however, for recruiting the help of other cells and molecules to destroy and dispose of pathogens to which the antibody has bound, and it confers functionally distinct properties on each of the various isotypes. The C regions of antibodies have three main effector functions. First, the Fc portions of different isotypes are recognized by specialized receptors expressed by immune effector cells. The Fc portions of immunoglobulin G1 and immunoglobulin G3 antibodies are recognized by Fc receptors present on the surface of phagocytic cells such as macrophages and neutrophils, which can thereby bind and engulf pathogens coated with antibodies of these isotypes. The Fc portion of immunoglobulin E binds to a high affinity Fc epsilon receptor on mast cells, basophils, and activated eosinophils, enabling these cells to respond to the binding of specific antigen by releasing inflammatory mediators. Second, the Fc portions of antigen-antibody complexes can bind to complement and initiate the complement cascade, which helps to recruit and activate phagocytes, can aid the engulfment of microbes by phagocytes, and can also directly destroy pathogens. Third, the Fc portion can deliver antibodies to places they would not reach without active transport. These include mucus secretions, tears, and milk, and the fetal blood circulation by transfer from the pregnant mother. In both cases, the Fc portion engages a specific receptor that leads to the active transport of the immunoglobulin through cells to reach different body compartments. The role of the Fc portion in these effector functions can be demonstrated by studying enzymatically treated immunoglobulins that have had one or other domain of the Fc cleaved off, or, more recently, by genetic engineering, which permits detailed mapping of the exact amino acid residues within the Fc that are needed for particular functions. Many kinds of microorganisms seem to have responded to the destructive potential of the Fc portion by manufacturing proteins that either bind to it or proteolytically cleave it, and so prevent the Fc region from working. Examples of these are protein A and protein G made by Staphylococcus species and protein D of Haemophilus species. Researchers can exploit these proteins to help to map the Fc as immunological reagents. Not all immunoglobulin isotypes or subtypes have the same capacity to engage each of the effector functions. And that's antibody C regions confer functional specialization. Have a great day.